Good morning. I'll go ahead and get started. My name is Vicki Fraser. I'm the chair of the Department of Medicine. It's my privilege to introduce the Robert Mendelsohn lectureship this morning. For those of you who don't have as much gray hair as I do, Dr. Mendelsohn was an esteemed master clinician in the Department of Medicine and Jewish Hospital for over 50 years here. So he did his medical training here, benefited from a Washington University School of Medicine scholarship, then went to the NIH for hematology research training and came back and practiced at Jewish Hospital for over 50 years. He was a regular attendee of Grand Rounds and used to sit up right in that top row and always had incredibly insightful questions. Long after he retired, he continued to attend medicine Grand Rounds. And we benefited from this lectureship because of the grateful contributions of his family members and also one of whom we have here, Dr. Nancy Mendelsohn, who we are thrilled to have join us today. So I was going to remind people that back in the day, it was not uncommon for physicians to bring their kids to work. And Dr. Mendelsohn benefited from her father bringing she and her brother to work and helping them find jobs in labs here. Probably a thing that we're not really able to do for teenagers so much anymore. But Dr. Nancy Mendelsohn is a distinguished pediatrician and geneticist. And she has a brother who's also a physician, scientist, and cardiologist who unfortunately couldn't be here with us today. So this lectureship really was inspired because of Dr. Mendelsohn's love of patient care and his commitment to compassionate bedside teaching and care of the patient. And he was a firm believer in the success of the care of the patient is focused on caring for the patient. He, when this lectureship was set up, the first lecturers actually were Dr. Nancy Mendelsohn and her brother, talking about their careers and their influence here at Washington University School of Medicine and their success as physician scientists. And then we've had really an outstanding group of physician scientists, in most cases focused on hematology and oncology. So today we are really very honored to have three speakers, the first of which is Dr. John DiPersio, who is an endowed professor in oncology, leader of the cellular therapies program, and a very successful physician scientist who has really led the division of oncology and built the division of oncology for more than 20 years. Growing the infusion business, the CAR T-cell therapy program, and the bone marrow transplant program. His research focuses on novel treatments and methods to improve stem cell recovery in order to facilitate successful bone marrow transplants, and also novel therapies to reduce the morbidity and mortality associated with graft versus host disease. John is an outstanding clinician, a really exceptional mentor and teacher, and a really brilliant physician scientist who has a number of patents and has led not one but 3 drugs to FDA approval, which are now in use, improving the lives of patients with leukemia and stem cell transplants. So we're very fortunate to have John speaking here as the first speaker, but we're also going to have Dr. Nichole Mercier and Dr. Leena Prabhu. So Dr. Mercier is the Assistant Vice Chancellor and the Managing Director of the Office of Technology Management for WashU. And she is responsible for leadership, strategy, and oversees all the operations of OTM here. She has made remarkable expansion improvements and improved efficiencies for physician scientists and PhD scientists to actually bring their discoveries to new companies, to patents, to technology that can improve human health. And then Dr. Leena Prabhu is the Director of Business Development and Licensing here at OTM. She and her team are responsible for evaluating all of the invention disclosures, managing patent portfolios, and negotiating licenses, and building relationships between scientists and commercialization business partners. So in the relatively short time that both Dr. Mercier and Prabhu have been here, they have really accelerated our faculty's ability to get licenses, to get patents, and to translate their discoveries into new businesses, new companies, new drugs, new vaccines, new treatments. And so we're really very fortunate to have the three of them tag teaming this morning. So please join me in welcoming Dr. DiPersio, Dr. Mercier, and Dr. Prabhu.
All right, so thank you, Vicki. You're the master of understatement, I'll tell you. Thank you very much for that kind introduction. So I don't know how this is going to work. We'll see. I fear that we'll be a little tight for time at the end, so some of the stuff I planned after Nichole speaks may be abbreviated, so bear with me. And this is a first try on this tag team approach. I first wanted to these are my disclosures. First time I ever saw this page in my life. There are 62 disclosures here, but actually only 5 of them are relevant, so I'm not sure where the rest of them come from. These are my true disclosures, and I wanted to jump ahead to a picture of Robert and John. John Mendelsohn is Robert's cousin, and I'll tell you why I'm showing this picture. Obviously, Robert is a family member of Washington University, and John is too. John is an esteemed and very famous physician scientist who really led the way in our understanding of targeting epithelial malignancies. And this is the two of them together later on in their lives. It's a great picture where they look alike, don't they? John Mendelsohn, I'll tell you why I'm mentioning John Mendelsohn. When I came here in 1994, I was recruited by someone named Stan Korsmeyer and Tim Lay and John Atkinson. They had asked John Mendelsohn who to recruit to run the transplant program here. And I had met John Mendelsohn when he was at UCSD and then at Memorial, and knew him well, and John recommended me for this position. So I'm actually thanking John posthumously for his recommendation that I consider a position here, which has been really probably one of the best decisions I ever made in my life, aside from marrying my wife, which I have to say in public. So John graduated from, I'll tell you a little bit about him. He graduated from Harvard in 1958. He worked in Jim Watson's lab as an undergraduate. He was a Fulbright scholar, and then went to Harvard Medical School, trained at the NIH, and then did his hematology oncology fellowship here at WashU. He was a faculty member at UCSD, where I first met him, and he worked with Gordon Sato, and he was the one, along with Gordon Sato, that developed antibodies to epidermal growth factor and HER2, and used these antibodies to block signaling in these epithelial cancers. The underlying hypothesis was that these antibodies would enhance the sensitivity of chemotherapy agents to apoptosis, which is obviously the foundation for many of the targeted molecules we use today for the treatment of all sorts of epithelial malignancies, really a giant in the field. He became the director of the UCSD Cancer Center, chair of the Department of Medicine at Memorial Sloan Kettering, and third president of MD Anderson Cancer Center in 1996. And again, was the person that really is responsible for me being here. I want to get to the subject at hand. The subject at hand is a project that we started in the laboratory, and it had to do with the treatment of a very rare disease called T-cell non-Hodgkin's lymphoma and T-cell acute lymphoblastic leukemia. And as you may know, there's been a lot of progress with B-cell malignancies with chimeric antigen receptor T-cells, but there's been essentially no progress with the treatment of T-cell malignancies, which are actually a devastating group of diseases with very poor prognosis and poor constellation of treatments that are available to patients. So looking at the outcomes of patients with T-cell acute lymphoblastic leukemia, this is a joint effort by ECOG, one of the cooperative groups in the United States, and the MRC in England, shows that the outcomes of newly diagnosed patients with T-cell acute lymphoblastic leukemia adults is similar to B-cell acute lymphoblastic leukemia. But when you relapse, you have only a 24% chance of survival at 1 year and only 11% survival at 6 years, even with the best available therapy. So obviously a devastating disease. So we wanted to pursue this, and we also wanted to stay away from the competition. There were hundreds of companies targeting myeloma and B-cell leukemia and B-cell non-Hodgkin's lymphoma, and we thought that this would be a disease where there's not much competition, not much knowledge, and maybe we could make an impact. So a very smart postdoc in the lab, Matt Cooper, started working on this, and this is the complications associated with targeting T-cell malignancies. The target antigen that you would go after in a T-cell malignancy is also expressed on a T-cell. So if you target that antigen, you would also target the effector cell that you put the CAR into. Thus, you would cause fratricide. So it would undermine the effort to kill the T-cell malignancy. The second is that the T-cells, these malignant T-cells are circulating in the peripheral blood, and there's no way to separate normal T-cells from malignant T-cells. So if you were to genetically modify the peripheral blood of a patient with T-cell ALL, you would also genetically modify the malignant cells, and thus you may make those malignant cells also resistant to the targeting that you developed. And finally, you could use somebody else's T-cells, but somebody else's T-cells are not matched to you, and they would get in the patient's blood and result in life-ending graft versus host disease. So our approach was to perform gene editing using CRISPR and to delete the target antigen, which is the target antigen we were going after in the T-cell malignancy, and the T-cell receptor alpha subunit, which is essential for the establishment of the T-cell receptor on the surface of a T-cell. This allows the T-cell to recognize one person from another person. This is a self-recognition system. And if you don't have the alpha subunit, the T-cell receptor cannot be assembled on the surface of a T-cell, and therefore that T-cell cannot recognize somebody else's body as non-self. And then you can put a CAR into that T-cell targeting that T-cell malignancy. So you had to do two things. Number one, in order to eliminate fratricide, you have to take out the target, the target you're going after, and also you have to take out the TCR. So this is multiplex CRISPR. This was the foundation of what we set out to do, and this is the foundation of Wugen, the company that we started here. So what target did we go after? Well, based on the expression of T-cell antigens and T-cell ALL, you can see that CD7 is the most overexpressed antigen in T-cell ALL, so of course we went after that. And there are a couple of other good antigens that are possible. One is CD2, and the other is CD5. So we actually have focused on both CD7 and CD2, and I'll show you that in a minute. So this is what it looked like. So we targeted CD7 because it's expressed on 98% of T-cell ALL. It's also present at high levels on a quarter of AMLs, which is a high population of patients. It's expressed on all these rare and devastating NK cell malignancies, and we also generated mice with CD7 knockouts, and so the T cells in those mice function normally. So we knew if we deleted CD7 in a normal human T cell, we wouldn't disable the T cell And then we generated chimeric antigen receptors, initially third generation. I won't go into all those details. It's not important. We were able to mark these genetically modified T cells with an epitope tag called CD34. And we used CRISPR gene editing to delete both the TCR and CD7 in the effector T cell. And then we put a CAR into these T cells targeting CD7 in the T cell malignancy. And that is a UCAR T7, which is universal CAR. That means my T cells that I harvest from my body and genetically modify them can be used to treat all of the people in this audience. So I could harvest enough of my T cells from one collection to treat everyone in the audience here with a T cell malignancy because they're universal T cells, and a single collection would be sufficient to treat 50 patients. There's more than 50 people in the room probably, but you can see that this is also economically quite viable as well. So this was an idea that we pitched, and we looked around, and this is where the choke point occurs. You have some nice science, published in a couple of nice papers, and then you're stuck. How do you bring this forward? And so Nichole and Leena will tell you a little bit about how this is done and how they facilitated here. But I can tell you from my perspective, this is a work in progress at Washington University. It is painful for everyone everywhere to do this. There are certain challenges with finding collaborators, finding supporters, finding venture capital. And it was so, I was very lucky in that I had a good friend that I had worked with for many years who actually was a very good scientist who was a principal at one of the local venture capital companies here in St. Louis, Rivervest, John McKern, and I turned to John McKern. Just fortunately, I don't think this could have happened without that being the case. And John, when we pitched it to John, he really liked the idea and then set up a formal review with his team at Rivervest. We pitched it to other venture capital companies like Third Rock, Atlas, 5AM Ventures, you name it, we did it, and they all sort of passed on it because they just thought it was too complicated and it wouldn't work. And so the guys at Rivervest here liked it because it was really targeting a unique group of patients. There was no competition, and it was a significant unmet medical need, and they liked the idea that and usually venture capital companies want to invest in reagents or technologies for which there's results of a randomized phase 3 prospective double-blind study. I'm being facetious, but they don't like to invest unless they know it's going to work. So the early investment is complicated. You know, sometimes early investments result in really no significant investment. Or if you take something further along, obviously the investment is more substantial and it is worth more. So with John's help, and then internally with Nichole and Leena, Rivervest went out, and John became the interim CEO of the company we started called Wugen, which was founded in 2018. So this is pretty amazing, since this was a company just founded in the idea in 2018. The first patient treated on a clinical trial was only like 2 years later. So that's world record pace. So usually it will take 3 to 5 years to get a patient treated on a new idea like this. So there were a lot of people working together, and there was a very complicated experience for me, and I think for even Washington University to get this going. So I want you to know how you name companies. So I came home one night after we got our first report from Rivervest that our Series A was successful and we had raised $30 million. And I said to my son, who was then this age, whatever that is, 8 or something, 7, 8, I said, Jack, Dad just started a new company. he said, Don, another company, Dad, your last company went bust. I said, well, that's true. So he said, well, what's the name of the company? I said, we don't have a name. And I said, he said, well, what does it do? I said, well, it slices and dices DNA. And he says, well, the greatest slicer and dicer in the world is Sensei Wu. And I said, Sensei Wu, W-U, Washington University, Sensei Wu. And I said, Sensei Wu. He said, yeah, that's the Lego master at Lego. We had just been to the Lego park. And and I said, Wu Genomics, Wu Gen. So that's how it got named. So that's how you name companies, basically. You just ask your kids. And I'll switch things over to Nichole, and she'll sort of review the OTM mission for you.
Thanks, John. Oh, good, you can hear me. So I have a Lego character that I proudly put up in my office of Master Wu. So I'm here to sort of be the boring part of the talk. It's much more exciting to hear about the science and where it's going, but what happens in the interim between when you make those discoveries and how a company like Wugen forms. So our office, for those who haven't heard of us, the Office of Technology Management, we are really, think of ourselves as collaborators. We have expertise, we know a lot of people. I tell people all the time, we have a lot of networks and I don't want to start a company. So what's the value of my network, but for all of you in the audience? So we want to take this expertise and the networks we have, along with the exceptional expertise you have in science, and really make things happen for the benefit of society. And we think that this actually enhances the mission of the university. So I know you are all out there creating new knowledge and disseminating that and training physicians and physician scientists. And what John described to you here is actually an extension of that. It is the creation of knowledge, and it is the application of knowledge. And we create real and tangible products. These came out of many universities, and we've had our own products that have come out. So many, many years ago, the test for heart attack was developed by Jack Ladenson. Precivity AD is a new product on the market for Alzheimer's. It's a blood-based a diagnostic test developed by Randy Bateman and Dave Holtzman, and many other people at the university. We have products out here. I talked about our goal is really to be a collaborator with you. We can't do it without you. If John doesn't want to start Wugen, I can't force him to do that. But I think it's really awesome that he wants to, and together I think we can make that work and we can use our connections. So we have quite a large team in our office, about 37 people, and we have multiple teams. I put 4, I'm pointing at the wrong one, up here. Our licensing team is what Leena leads, licensing and business development. We have a whole group in our office that can really help you understand how do patents happen, what is a patent, how do you think about one, and how can you create in the space you're in. It's not for today's talk to go over a patent, but suffice it to say, before you go out and publish something, you should talk to us, because it can impact your ability to get a patent if you go out and talk at a conference or you publish a paper without that. And then we have a group that will help with technology development, because a lot of what John talked about in that basic research, it needs to be attractive to an industry partner or an investor, and there's certain ways to set those questions up in a way that would attract industries or investors in a more probable manner. And so we have people in our office that think about that, and then we have our new ventures team, which helps stand up companies, which John did not have the benefit of at the time. So I'm not going to walk through this slide, but this is our process. It starts with an invention disclosure. That submission happens through our portal, or you can just reach out and contact any of us and we'll help you with that. We have a lot of things that happen in each of these buckets, right? So you can think of disclosures and patents and licensing as sort of like the 3 pivotal points, but there's a lot that happens in between that, because I know when I was trained in my doctoral degree, I did not know what a patent was. I didn't know we had a tech transfer office. There's a lot we just aren't exposed to. I suppose now you're exposed to more than I was many years ago, but it's okay if you don't know. We all started in that place. And then there's programs to really help advance, programs and funding to help you advance the asset. So you might think, I don't really have the money to be able to do that. We can help you with that. So this is the problem, right? You're all here in this basic research, you're doing discoveries, and then while I have money for that, and I'm not going to talk about the current environment. Hopefully we continue to have money for this basic research. We're trying to do a lot of education in this space so that you understand this entire process, but this valley of death is real. It is hard to find the money to prototype. It is hard to find the money to advance the asset. And so that's where our partners come in, and that's where we have our ventures team, and we have money within the university. We have the Needleman Fund that John actually has money out of for another project. And we have a Center for Drug Development. So there is money to help you advance assets at the university, and then our office also has a small fund. Now, what John didn't have access to when he started was a group and a team that could really help him put together what is the opportunity that an investor would invest in. And so now I have 4 people on our new ventures team. One of them has pharma experience, another ran a medtech accelerator. We just hired somebody on physical sciences, and then we also have a biomedical engineer now, so 4 people. And when we started our new ventures group, we looked at at that time it was just about 100 companies that had come out of WashU, and we looked at all of them. We said, what makes it successful? And our definition of success is it exits. So that means there's a merger or an IPO or something like that. And when we looked at those companies that were successful, they all had 2 things in common. They had dilutive investment from, could be a venture capital, could be an angel, seed fund. There's lots of ways to get dilutive investment. They had that within 6 months, and they had an external CEO. These companies were more likely to get to the exit and to receive follow-on funding. And so that was the thesis when we stood up our new ventures team. We're going to stand up those types of companies. And you can see over our entire history, this is the number of companies that started with dilutive investment at the outset. And some of these years, so 1 or 2, some in 0, that's not good. So this last year, our new ventures team has been around 2 years. I guess this is our 3rd year. Last year, we started 10 companies. 50% of those had dilutive funding at the outset. We are poised to probably start 8 to 9 companies this year, with more than 50% of those having dilutive investment at the outset. So that's where we're going. We're happy to talk more about that. But this is really the key. So we go to partnering meetings. We go to JP Morgan, which is the biggest investor partnering meeting. We go to the bio meeting, many, many meetings. And so we know investors. This is just in 2 years how many investors we know and how many we can get to and how many who read our emails, essentially. This is the number of pitch decks we've circulated. So our new ventures team, when they work with you, will really sit down and try to understand your science, start with a scientific deck, and make it attractive to an investor. So we call that an investor-ready pitch deck. We have now had many, many of our research teams giving pitches themselves to venture capital. And I know you probably think, well, 70, well, that's in 2 years. So we are really advancing the number of our faculty and research teams who are engaging investors, and you learn more about how to do that engagement the more pitches you give. And then we're tracking this dilutive funding piece. So I think, again, I think we want to be the partners. We want to help Wugen even more accelerated than maybe John, maybe lower the barrier for the work that John had to do and go out and talking to investors. And I think, okay, so one other thing John mentioned when we were sort of talking about this talk, what are the challenges that we face in startups? And I think, again, we've tried to address this with our new ventures team. Having the external business person negotiate the license with the university is really important. It takes that off of John, so he does not have to do that. He can focus on science. We maintain a really good relationship because as business people, we're comfortable with conflict in a way that I think you only get comfortable with that the more you do it. And we know we're never going to walk away from a deal because we want to make it happen, but we also want to get the best deal we can on behalf of the university, on behalf of John. So having an external person be able to negotiate, that's really important. someone who understands the financial terms and what that means for the company and where the trade-offs are is really important so that they can align with how they're developing the early stage company, align that with the university as well. Because there's always going to be a collaboration between the research team and the startup company in those early years. So it's very important that we're sort of all aligned in this, that there's an arm's length transaction. And we know that startups have a high failure rate. John's son told him that, but we're super optimistic about Wugen. But again, sort of setting it up correctly from the beginning can mitigate that, because the biggest thing that where companies fail is because of the team. And we would love all of the companies, like all failures to be due to science, but they're not all due to science. Oftentimes it's due to how the company was set up and how the team came together. And again, it's a long-term relationship. You will wear two hats. You will be the company person and the university researcher. You'll always have that, and you'll have to navigate that conflict for yourself. And Leena and I navigate that conflict as well, and we're trying to help you understand which hat are you wearing in which conversation. So I'm going to let Leena talk more about challenges here.
That is contemplated. For example, if you look at gene therapy studies, they are at the higher end. A typical phase 1 clinical study runs in millions. When you look at the annual FDA approvals, in recent times, it's about 50 to 53 new therapeutic assets that are developed and approved. And out of these assets that are approved each year, oncology is approximately about 25 to 30%, neurology about 15 to 20%, infectious diseases about 10 to 15%. And as you see the assets being progressed from stage 1, which is basic research that happens in your lab, to maybe a stage 2, which is drug discovery, screening, you see this is an inverted funnel. As you advance, there are fewer and fewer new chemical entities that are moving forward. This is a little more granular, where you have assets going into a phase 1 clinical study. Of the total number of these assets that go into a phase 1 clinical study, about 10% to 15% advance to a phase 2. And of those, about 20 to 30% are advanced to a phase 3 clinical study. And then the likelihood of getting an FDA approval is enhanced tremendously. About 50% of these NCEs, new chemical entities, would get a FDA approval. So you see this is a high risk, high reward enterprise and a process, which is why increasingly a lot of university tech transfer offices are wanting to bridge that gap, the gap between basic science and when to get a commercial entity to the table to take a license. The more you de-risk these therapeutic assets, the higher the chance of attracting the right kind of partner that John mentioned in his talk. Like Nichole mentioned, when you form a startup, you as a startup co-founder are going to face some conflict. This conflict can be effectively managed because the goal of the university COI policies is to ensure that patient care and research that's going on here is perceived not to be compromised by, personal gain that could arise from having equity provisions or licensing royalties in a license agreement. The COI office is a separate office, but we do work closely with that office in empowering our faculty co-founders to navigate this space. Let's now take a quick look at the WashU therapeutics portfolio. We have about 10 assets in the preclinical stage, like I mentioned before, about 3 in phase 1, 3 in phase 2, and we're expecting some of these to advance into phase 3 and FDA approval process. On the market, we have Covaxin, which is a nasal COVID vaccine that is being marketed by Bharat Biotech. This technology came from Drs. Diamond, Mike Diamond, and Dr. David Curiel.
But let me just tell you a little bit more about the focus of this particular company. It's, again, it's a company focused on the use of NK cells, driven largely by Todd Feniger's laboratory. And these activated NK cells called simul NK cells and CAR T cells, which I've just mentioned. So this is our contribution, the use of universal CAR T cells to treat T-cell malignancies, and Todd's contribution is a memory NK platform. Both of these are being developed coincidentally at Wugen. As I said, the first patients were treated on a phase 1 study shown here. The lead investigator, after we worked out the conflict of interest issues, which were Herculean, I thought, and really many people take credit for getting this done. So I thank all of them for all of their help. But this was not easy, and Armin was the PI of this. This paper was just accepted in Blood, and it's a phase 1 study which led to the pivotal trial which Leena just mentioned. This is sort of a snapshot of a single patient. You can see that this patient before treatment had disease all over the place, and a patient actually referred from the Mayo Clinic with refractory T-cell ALL who had gone through, I think, something like 12 or 13 different treatments and was dying, and then 30 days later had a complete response. And this just shows that this therapy is efficacious. It's not without its toxicities. This is the result of the phase 1 study. So 82% of the patients had CRs that were MRD negative at day 30. And so the overall composite CR rate for the entire phase 1 study was around 75%. And again, that suggests high efficacy for a group of patients that had no other option for treatment. I mentioned that we wanted to also test this in T-cell non-Hodgkin's lymphoma and AML, and Mike Kramer, who was a fellow at the time, now a junior faculty member, and Jeff Wee, who's the director of the transplant program, wrote a nice study testing this. And in fact, this study was opened. It's currently closed because of lack of reagent right now, because they're still making more reagent, and also there was some toxicity issues. But that study eventually will reopen, and we'll be able to test this in AML and T-cell non-Hodgkin's lymphoma. So this is the announcement for the registration study. So this is a big deal, and hopefully this study will complete. And if this study is positive, there would be this would be the foundation for FDA approval if the drug continues to show the same level of efficacy and a tolerable level of toxicity. So what's next? So we're doing a lot of other things, and each one of these things, I'll go through this very quickly, each one of these has to involve translation to the clinic. So that means finding partners, developing preclinical methods to develop these technologies, to find venture capital, to work with OTM, to work out conflict of interest issues. So there's many things going on, and each one is amplified every time we take up a new project. This is the first project I'll mention, and this is work done by Jerry Zhang and Mike Rettig in the lab. And what we thought is that we found that in the T-cell ALL study, a number of patients, of the few patients that relapsed, a number of them had CD7 negative disease. So we knew that they lost their target antigen as a way of immune escape. And so we thought looking at a second antigen, which I mentioned before, CD2, would be a way to approach these patients that relapse. And so Jerry and Mike have worked out a really elegant way of targeting CD2. But unfortunately, when you take CD2 out of a T cell, you disable a T cell. So Jerry's figured out how to actually mutate the endogenous CD2 so it's invisible to the CAR to CD2. So we made a CAR to CD2 which cannot recognize the mutated CD2 that is in the T cell. So this is called epitope masking. So in other words, you can target the T cell malignancy effectively, but not result in fratricide, and still produce a CD2 in the effector cell that is functional. It's kind of incredibly clever, and we're getting to the point now where we're looking for partners. And we are close, I think, and with OTM's help and with Conflict of Interest Office and the attorneys here with their help, hopefully we can move forward, and we'd like to actually be in a position to carry this through to a phase 1 study, and then if it's positive, our partner will have the first right of refusal for licensing the product. So that will allow us to do the research here, that will allow us to write papers, that will allow us to treat patients without issues of conflict of interest, and then we can license it to our partner who has the first right of refusal. So I won't go into too many details, that's the basic fundamental issue, and this is based on a number of very beautiful papers published by other groups looking at epitope masking as a way of targeting these targets. And I'll skip through this because we don't have that much time. I wanted to mention one other thing. Obviously, if you're targeting T-cell malignancies, you will not only kill a T-cell malignancy, but you'll kill all the normal T-cells. And so Miriam Kim in the lab, who is now an independent investigator focused on CAR-T and myeloid malignancies, published a nice paper in JCI Insight in which she actually deleted the target antigen in the stem cell population, and so she was able to show that in a mouse model that when you look at those stem cells that have the target antigen deleted, in this case CD7, that those hematopoietic cells that engraft in these mice are CD7 negative but are normal in numbers of T cells and immune cells. So these mice have normal immune function, but they're resistant to targeting because the CD7 can't see them because they don't have CD7 on their surface. So this is a way of getting around really the major problem with targeting T-cell malignancies with either UCAR-T7 or UCAR-T2, and that is the persistent pancytopenia, but more importantly, the persistent immune dysfunction seen in these patients and the terrible high frequency of life-threatening infections that we have seen and also other groups have seen that target T-cell malignancies. And so I'll just go through this. The third is to sort of get these CAR-Ts to work better, and this has been an effort to work with several companies, and we've patented ways of enhancing the use of certain cytokines to enhance the expansion and persistence of CAR-Ts. And I'll just show you one example. This is a long-acting interleukin-7, so we give CAR-Ts to mice that have tumors. That's the light that's being emitted from these guys. And you can see the CAR-T actually provides some benefit. This is tumor in these mice, but when we give a couple of doses of the IL-7, the tumor goes away and all the mice are cured. And so this is a way of actually markedly enhancing CAR-T cell expansion, and this also is something that we're working with another company. We patented, and we're trying to figure out how to take this forward. And this is more data showing the same thing. That was in a T-cell tumor, this is in a B-cell tumor, and this is in a myeloid tumor, all of which show the same thing. Just a couple of doses of this cytokine can convert a relatively ineffective CAR into a completely effective CAR. And we are translating this into the clinic. And so this is the other benefit of doing this translational research, is that you can engage all of your junior faculty. They can write grants, they can write papers, it provides a lot of vigor to the academic environment, and people that are not necessarily hardcore bench scientists can get involved in the work. and get excited about what they're doing as important translational work. So this is work done by Zach Krees and Armin looking at testing this IL-7 in the clinic. We finished this study. It looks very good, and we have the next study, which is going to be starting very soon, which we're funded for. And so the last is to control the toxicities of the CAR. So I told you about, getting rid of the immune-suppressed environment by deleting the target and the stem cells and enhancing their expansion and persistence by using certain cytokines. And so the one thing that these patients can die of is something called cytokine release syndrome, which is this mysterious effect that they get fevers, capillary leak, sometimes neurotoxicity, and they can die from this. So we want to see, and this is particularly the case in targeting T-cell malignancies. So we want to see if we can fix this. So there's a whole group of people in the lab and other people that we're working with outside of the lab to see if we can reverse this. And we've taken two approaches. We've done high throughput small molecule screens to see if we can identify kinase inhibitors and other small molecules that block the production of interleukin-6, which is the surrogate marker of cytokine release syndrome, without affecting CAR T-cell function, and we finished those. And we've also performed CRISPR screens looking at which pathways and genes are involved in the production of these cytokines that are inflammatory and that we think cause cytokine release syndrome. So we've actually done a lot of this work, and we've identified a couple of targets, and we're taking them to the clinic. And so this was the first study we did, and we showed that interferon gamma was one of the key mediators of this, and we knew that JAK inhibitors block gamma interferon signaling and production, and so a randomized study was done with a JAK inhibitor as CSF, as CRS prophylaxis, and this is the results of this. This was just published in Blood, and you can see that, and we were the driving force in the laboratory in the main authors of this publication, and so this is the patients that did not get itacitinib, which is a JAK inhibitor, and these are the patients that did, and this is the incidence of grade 2 or greater CRS, and so you can see that itacitinib is very effective at reducing CRS in these patients. So that's one approach. And then internally, we identified an inhibitor called dufulisib, which is a PI3 gamma delta inhibitor that blocks CRS in our assays. And this also was tested in the clinic. And this study is now done and ready to be published and showing the same effect. And when we put these two drugs together in our assay, they were synergistic. And so the future of CRS mitigation is to combine both of these drugs together. And we showed in both cases that the drugs do not affect CAR T-cell function. And so my conclusion is that that these universal CARs to 7 and 2 are effective at killing T-cell ALL, cell lines in vitro and in vivo. This UCAR-T7 is a very effective treatment for T-cell ALL and T-cell non-Hodgkin's lymphoma in the clinic, but we have toxicity issues that we need to manage. NTI7, which is this long-acting IL-7, can dramatically enhance expansion, persistence, and anti-tumor efficacy, and may have general applications to all CAR T-cell therapies. We just have to make sure that this IL-7 does not also enhance CRS. Epitope masking is a new way of actually approaching many hematologic malignancies for which there was no other way to target in the past. And I think this provides an incredible potential approach for targeting lots of things that we didn't think was possible in the past. We just have to make sure that masking is effective, doesn't alter the function of the genes we're trying to mask. And then it's also, part of this is also it has to be coupled with a stem cell transplant. And then mechanisms of CRS, which are still murky, are slowly being unraveled, and we have good ways to mitigate CRS now and treat it. And I think that I can say that due to some of the work done here and in other institutions, and some of the translational efforts that have been really helped by a really large group of people here, investors, OTM, investors in the laboratory, and the patients that participated in these trials, that there is now hope for treating T-cell, relapse refractory T-cell malignancies with CAR-T and other immunotherapies. And I'd like to thank all the people that did the work and contributed to this, especially the bolded people, Mike Reddick, Jerry, Miriam Kim, Julie Ritchie, Parmeshwar, Rekha, people in the Division of Oncology mentioned here, Zach, Mike Slade, Mike Kramer, Jeff Wee, Armin, Wugen collaborators, OTM, critical staff and collaborators in OTM, Neoimmune Tech, Rivervest, Secura Bio, and Insight, and there are many others. So it's a challenge to put all these these cats into one corral and get them to go in the same direction. But it's been an interesting journey for all of us, and I want to thank you for your attention. We'll take some questions.
Well, that was a terrific talk, a tour de force of a lot in a short period of time. I think we have a few minutes for questions. John, if I may start, why do only half the patients get CRS, and is there a way to predict who's going to get it and who doesn't?
Yeah. So if you look carefully enough, probably around 70 or 80% of patients get some kind of CRS. Only about half the patients get grade 2 or greater CRS. So T-cell activation is a sine qua non of CRS. So T-cells get activated near or in direct contact with a myeloid cell. The T-cell communicates with a myeloid cell. The myeloid cell makes all the bad stuff. So it's not the activated CAR T that does it, it's the myeloid cell, which is an innocent bystander that makes all the bad stuff, and that's the CRS. The more active the T-cell activation is, the more significant the CRS is, because the myeloid cells get engaged. And the myeloid cells can be primed, and so there's probably some contribution by things like infections, bulk disease, that contribute to the overall level and quantity of CRS.
Great. Other questions?
John?
Yes.
Nancy?
Congratulations on your accomplishments. Can you speak to the patents and their duration? I think probably some of your early work was patented and protected, but I'm expecting the patents are probably getting closer to expiration. Is that an issue, or just the barriers to entry so great that the patent position isn't that important? Can you speak to that?
Yeah, so I think our patents are good for a lot of years. I think we issued, 20 years. So I'll be dead. So I don't really care what happens after I'm dead. Rich. So we have a lot of patent protection. And that was the other thing that was amazing to me. So I don't know if anyone has met patent attorneys. They're a different bunch of characters. There's like it's crazy, it's amazing what they can do. They know everything. They put this stuff together. Yeah, these are the kind of people that you can't live without, because professional people that understand how to put patents together, working with institutions, OTM, but they're magical in what they can do. And they just keep on turning them out, because you got to cover everything. It's quite a chore, actually. And I had no idea, but we use, who do we use, Posanelli mostly, or, yeah, so. Yeah.
Nancy?
One quick question. I think the OTM model is fascinating. And do other universities do this, or is this unique to WashU?
Yes. So other universities, most major research institutions have tech transfer offices. They're all set up differently. They all offer different things, and some are more robust than others. But I would say over the last decade, we've tried to adapt to be a better partner with WashU faculty.
Yeah.
Russell?
Great talk by everyone. John, as kind of the inventor and so on of the company, as it's progressed and you've been less involved in the company, how have you felt about kind of losing some of that control over the initial science?
Yeah, so you know my personality well, Russell. So there's two things that can happen when you start a company. One is that you can be a part of it, really day-to-day part of it. I think that is really complicated, because it takes time away from all the other things you're doing. So I actually decided not to do that, and it's simpler that way. You just do your lab work, and then the company goes on. But you think that you're going to have some role, but you're deluding yourself, because once you step back and license it technically, they hire the CEO, they hire the chief scientific officer, they hire the marketing people, they're them. And then you might say to them, you need to do this, and they would say, oh yeah, that's a good idea, but then they turn around and say, that's the stupidest idea I ever heard of, we're going to ignore this guy. So you're really out of it. So I think that there's two ways to do this. You either stay in it at some sacrifice to your time and effort, and there's also conflicts there, or you step back. And I think if you can step back, it's probably a better model. I'd like to step back, but also like to have the option of testing the drug that you've developed in your own institution. That's the thing that's called management of conflict. That's what we have to do. We have to be able to develop reagents and test them at our institutions. Why not? If you have a drug that might save someone's life, why does it have to be tested elsewhere? Why can't the patients in your region have access to it? So that's the kind of thing. So you just have to make these firewalls internally solid enough so that you don't get stuck in, consenting a patient or caring for the patient. If you can do that, I think that's the key. But I prefer the model where you start something, let the company do it. Maybe you can provide them advice as a scientific advisor on the scientific advisory board, but there's two ways that you have to say, you stay in it or you stay out of it, and I've chosen to stay out of it.
John, Nichole, Leena, thank you so much.